Hello everyone, my name is Alexis Joly. I'm a computer scientist at Inria in France, and I'm going to talk about new fine-grained visual classification challenges related to biodiversity. So first of all, I would like to thank all the people who contributed to this work, and in particular the members of the PlantNet team, of which I have the pleasure to be the scientific leader. So my topic today will have links with the PlantNet platform, but it will be somehow more general since the challenges presented here have a broader interest in the field of biodiversity monitoring. So to begin with, I show here a global vision of what could be a biodiversity monitoring system on a continental scale. And you have as inputs the data acquisition part, which is composed on the one hand of field observations of the species, such as citizen science data or expert data, and on the other hand, remote sensing data or environmental rasters. And from this different data, the objective is then typically to generate high-resolution maps of species and their habitats, and then to develop applications that allow decision makers to make better informed decisions regarding biodiversity. So in this context, we have been developing since 2013 the PlantNet platform, whose main originality is to rely on machine learning to help people identify plants with their smartphone and then contribute to their inventory. So nowadays, the platform has become one of the largest citizen observatories in the world, with nearly 10 million contributors collecting geolocalized plant observations in more than 200 countries. And as we will see in the rest of my presentation, this data is of high interest for biodiversity monitoring, as it is actually already used by many researchers in international platforms such as GBIF. So, however, I will show in this presentation that there are still many challenges to be solved and that some of them also represent great research opportunities for fine-grained visual classification. So the first challenge I would like to present is about set-valued species identification. So as you may know, there are nearly 400,000 species of plants on Earth, and it can be very difficult to distinguish them from each other. In particular, if two species have the same leaves but different flowers and you only photograph the leaf, you will have an irreducible ambiguity that you will never be able to remove unless you go back to see the plant itself. And from a theoretical point of view, this type of uncertainty is called an aleatoric uncertainty. And its particularity is that even the optimal predictor will lead to a significant classification error. And thus, in this case, it seems preferable to predict a set of species rather than a single species. And this is somehow the only way to reduce the error rate if you don't have additional information. Considering only the set error rate, however, is too naive because the predictor that always predicts all classes is very accurate, but it is completely uninformative. And thus, this accuracy needs to be balanced with a measure of informativeness. There are several ways to define this informativeness and to measure the trade-off with the error rate. And this gives different formulations of the set-valued classification problem, which are adapted to different scenarios. And the one we are focusing on here consists in controlling the average size of the predicted sets. And this can be seen as a generalization of the TOPK classifier to sets of various sizes, but still equal to k on average. A baseline predictor for this problem is to simply threshold the probabilities estimated by the model, so typically the output of a softmax. However, we believe that there is plenty of room for alternative approaches that would allow to learn a better predictor. And therefore, we have been releasing a new dataset built from PlantNet database, and that consists of 300,000 images covering about 1,000 plant species. And the main originality of this dataset is that it was constructed by retaining only a subset of the genera of the entire PlantNet database, while retaining all the species that belong to those genera. And doing so, is intended to preserve the large amount of ambiguity present in the original database, as species in the same genus are likely to share common visual features. And as a result, the dataset has both a high level of ambiguity across classes and also a very strong class imbalance, as in any biodiversity data. So we have been training a baseline ResNet 50 on this dataset, and as you can see here, the mean of the top K and average K accuracy over all the species in the test set is actually very low. And to encourage research on this problem, we submitted this dataset to the datasets and benchmarks tracks of NeurIPS 2021, and we hope this will foster research on this set-valued classification problem. So now the second challenge I would like to present to you is also related to the problem of plant identification, but is more specifically concerned with herbarium data. So as you may know, there are hundreds of herbarium collections in the world, and many of them have already been digitized, so that there are now millions of herbarium sheet images available, especially for the least frequently photographed species, which are also de facto the most difficult to recognize. And the question is whether this herbarium data could be used in a complementary way to photographs for training identification models used in mobile applications such as PlantNet. And to answer this question, we organized a dedicated challenge within LifeClef. It is essentially a cross-domain classification task, but it is very difficult because the visual aspect of the herbaria is very different from the field photographs. The specimens are indeed dried and pressed before being mounted, and in addition, many of them are very old, so that they have undergone important changes with age. Concerning the evaluation methodology, we shared 3 datasets with the participants, so one large-scale training set composed mostly of herbarium sheets, one validation set composed of specimens in both domains, and one test set composed solely of field pictures, and for which the trained models had to guess the species names. So I show here the results that have been obtained during the 2020 evaluation campaign. So the first main outcome was that a classical CNN trained on the available training data does not work at all, and even when external field pictures were used as complementary training data. And the only methods that managed to obtain encouraging results were those that relied specifically on domain adaptation techniques. So in particular, the method that obtained the best results was an adversarial domain adaptation network that was extended so as to take into account the genus and family levels in addition to the species level. The second best method was rather based on a triplet loss network, and it performed lower on the whole test set, but interestingly, it also performed much better on a subset of the test set composed of the most difficult species. That is, those with very few or no field photographs at all in the training set. So overall, however, the performances are still quite low, and progress must be made on these methods if they are to be integrated into PlantNet one day. The third topic I would like to address now is a bit more reflective and concerns the filtering and cleaning of the raw data that is collected in a collaborative platform such as PlantNet. So if we look at some figures about PlantNet data, we can see that out of the 300 million plant observations collected, only 4 million have been revised in a collaborative way, thanks to the different mechanisms set up in the applications. And manual review is actually a process that requires time and user involvement, and it cannot be expected to be sufficient to review all data. And thus, in addition, we need to implement automatic processes for filtering and categorizing data. The first problem is the presence of noise in the data. So you wouldn't believe how creative some people can be, let alone people with bad intentions. And some of this inappropriate content is fairly easy to filter out, as is the case with the examples on the left of my slide, for example. You just need to add a supervised rejection classes to your recognition model and enrich it over time. Some images, however, are more or less complicated to process, such as the ones on the right. In these images, you already have a plant present in the image, but in addition, you also have other objects or people. So this is a multi-label classification problem, but it is complicated by the fact that you usually only have presence labels for the subjects and no absence labels. And moreover, it is difficult to determine which images you want to keep or not, even if you detect the objects present well. It will indeed be necessary to capture a notion of importance of the plant in the image or of intentionality of the user. A second problem is the heterogeneity of the data. So since PlantNet is used in a variety of ways by a variety of users, we also collect various types of content, for instance, house plants on one side and wild plants on the other side. And the problem is that when we want to answer a particular scientific question from PlantNet data, we have to use only a certain type. For instance, ecological niche modeling requires only wild occurrences, whereas you may need several types of content for studying invasions of alien species from gardens. Here again, the problem of classifying these observations according to a given typology can be more or less difficult. Some categories are fairly easy to recognize, such as houseplants or captive plants at the top left. Other categories are more difficult to recognize from the image alone, but they could be more easily recognized by using other types of information, such as the geolocation and some land cover data. This is the case, for example, for the detection of plants growing in or next to crops, which are of great interest for studies in agroecology. And finally, there are some categories that are very difficult to detect automatically, although they are no less important for data analysis. And the distinction between plants managed by humans and spontaneously growing plants is, for example, a very important piece of information that is part of the Darwin Core Standard, for instance. And as it can be seen in the images on the right, automatic annotation of this type of label from images does not seem to be a trivial problem. And we will probably try to build a training data set in the next few years to evaluate this. So for your information, I present here the automatic processes that are currently implemented to filter PlantNet observation before they are shared with GBIF. So some are AI-based, in particular for validating the species names and for removing potted and cultivated plants. And another one is knowledge-based and is aimed to remove all observations of plants that are known to occur that are not known to occur naturally at the location where they were observed. And with these filters, we already share many more observations than just the manually reviewed data, but the recall and accuracy of this filtering could be greatly improved with more advanced fine-grained classification models. So last but not least, the fourth challenge I would like to address is the prediction of species distribution. Knowing which species live in a given area is indeed an essential issue, whether for land use planning, for instance, to give permission for the construction of new buildings in a natural area, or for conservation or environmental education issues that require detailed knowledge of the local biodiversity. And to determine the species present at a given location, one will of course rely to a large extent on existing occurrence data, such as those of the GBIF. But this is far from sufficient because there are far too many areas with no occurrences, and there are also many species with very few occurrences. So what ecologists do to solve this problem is typically to combine this occurrence data with environmental data, such as rasters of average temperature or elevation maps. And this allows them to model the environmental niche of each species and then to use these models to predict where the species are likely to occur. And in our current work, we therefore ask whether deep learning approaches could allow us to build species distribution models with a much higher predictive power than those used by ecologists. And the use of images as inputs to the model in particular is one of the strengths of deep learning, whereas classical ecologist models are not able to capture such complex information with a spatial structure. So the models we have trained so far are typically convolutional neural networks that take as inputs multi-channel images, such as remote sensing patches or environmental patches, characterizing the environment in a given location. And then the model predicts as output the species that are the most likely to be found in that location. So to evaluate this type of approach, we have built, in collaboration with Caltech and iNaturalist, a data set of several million plant and animal occurrences, each matched with a multimodal image of 1 meter resolution around the occurrence and composed of both remote sensing and environmental data. So this data set is shared in the context of a challenge called GeoLifeClay, part of FGVC, and about which I invite you to attend the presentation of Titouan L'Oriol later in this workshop, who will make a synthesis and present the results of this year. In the meantime, I would just like to comment a bit on a question that has been asked about this challenge, and that is whether or not it is truly a fine-grained visual classification challenge. And to do so, I suggest you look at this TSNE visualization that was built from the features learned by an Inception V3 model on a sample of the GeoLifeClay test set. And what we can see at first sight is that the model did not separate the species into distinct clusters, as we can see in some classification tasks. And this seems quite logical, since we cannot really distinguish a particular species from the input images, of course. But on the other hand, if we look in more detail, we can see that the model has captured very well a notion of habitat of the predicted species, with on one side mountainous areas and on the other side urban environments passing by a variety of environments such as forests and agricultural areas. And to understand even more finely what the model learns from the images, we have realized here another visualization built from a three-dimensional TSNE projection of a set of occurrences located on a grid of a few kilometers side. And here it is the color that encodes the features learned by the CNN, and we simply displayed the color of all the patches of the spatial grids. And this allows us to see that the model is in fact able to distinguish very fine visual patterns, such as different types of roads, different types of crops, different types of forests, or different types of water bodies, such as lagunas, lakes, or ponds. And so personally, I would think about it as a fine-grained classification problem, even if the objects to be recognized are not directly the ones provided in the labels. So to conclude, I hope that the different challenges I have presented today have given you the desire to continue working on these problems of fine-grained classification applied to biodiversity. And I thank all the organizations and companies that have contributed to the present work. And thank you for your attention, and goodbye.